Hi, everyone. Thank you so much for joining our Tech Exchange series. It is my pleasure to introduce today's speakers. Our first speaker is Dr. Saptarshi Chakraborty. He's an assistant professor in my department, biostatistics, in the School of Public Health and Health Professions. He's also an affiliated faculty of IAD. He has many accomplishments and roles. He's the head of professional advancements in biostatistics, epidemiology, and research design, which is a core of the CTSI Center at UB. And he's also serving in the IBS Biometric Bulletin correspondent for Eastern North American region as an associate editor of the Indian Journal of Statistics. He received his PhD in statistics from the University of Florida in 2018, where he went on after that to do a 2-year postdoc in statistical genomics at Sloan Kettering Cancer Center in New York before joining University of Buffalo in 2020. So today he's going to talk to us about topic models for mutational signature analysis.
Thank you, Rachel. Thanks for the very nice introduction, and thank you for having me here today. It's a pleasure to give a talk in the Tech Exchange seminar series. As you can see from the title of my talk, it's going to be about topic models for mutation signature analysis. In today's world, natural language processing, particularly large language models, have been a key. We all know like how promising the technology is. My today's talk is going to show about some other areas of NLP and how we can successfully utilize them in many other areas of contemporary science. This talk is a combination of some of my recent and ongoing work done in collaboration with my colleagues. First is Shuangcheng Hua, who is a former student of mine, PhD student, received her PhD last year, and she's now a biostatistician and AbbVie, along with my collaborators, Zoe Guan at Mass General, and Colin Begg and Rong Laishan at Memorial Sloan Kettering Cancer Center. So I'll start with, just to set this stage, I will start with a very high level, very simplified version of the biology that we will need to get an understanding of what the problem is. And at a very high level, we all know human cells have nuclei inside which there are these thread-like objects called chromosomes, which pack the human DNA. For the purpose of this talk, human DNA, I will treat it as a long chain of nucleotides on which there are regions. For the purpose of this talk, I will treat those blocks or regions as gene. The premise of my today's talk is that due to various external factors like exposures to ultraviolet, smoking, alcohol consumptions, and other factors, alterations happen in the DNA. Alterations happen all the time. There are some particular factors that lead to specific types of alterations. These alterations are called mutations. So mutations are alterations happening in the DNA, possibly inside these specific blocks or regions called genes. Somatic mutations are mutations that happen after conception and are not carried forward to the next generation. And the premise of my today's talk is that a large and growing body of research has evidence that specific somatic mutations are strongly related with cancer. Specific areas on the chromosome that get mutated are strongly associated with different types of cancer. So the goal in this line of work, a key area of in contemporary computational biology and computational biomedicine, is the goal to characterize different cancer types and predict cancer risk based on somatic mutational fingerprints. Somatic mutations cause cancer. So the idea is, based on the genomic fingerprint, are we able to comment anything about cancer, the possibility of cancer? There is numerous clinical applications of this paradigm. The first is the problem of understanding primary site of origin of a metastasized tumor. So cancer metastasizes, it translocates into different parts of the body as the stage progresses. Now, when cancer metastasizes, the tumors appear in different parts of the body. They are extremely heterogeneous in terms of, like where they appear, but there is some genomic similarity, some genetic similarities happening within the tumors. The idea is that if we are able to connect different cancers with the genomic fingerprint, we might be able to analyze the metastasized tumors, look at its genomic fingerprint, and then try to predict or connect what the primary site of origin is. Because in later stage, if we are able to understand what the primary site of origin is of those metastasized tumors, we are going to be able to have better prognosis and treatment options. Prognosis of rare infrequent cancer is a related problem where cancer happening at a small organ on which we do not have enough data from previous databases. Third is the modern holy grail of research, liquid biopsies, understanding the probability of having cancer early in the process. Somewhat similar to diabetes test, where we aim to do a blood test, and from the blood sample, we want to see if we get any tumor fragments that will give us early alarm for certain types of cancer. It's a very heavily, it's a highly active area of modern research. Finally, fourth example is predicting cancer risk based on specific risk factors. This is also a very active area of research. Exposure to certain environments or certain types of infections may lead to specific types of cancer. All these things, the underlying framework is that if we are able to connect the genetic fingerprint with cancer at a genomic level, we might be able to predict the probability of having a specific type of cancer down the road. And there are several other related clinical applications for this framework. The statistical and machine learning goal of this is to build a classifier or a predictive model that's going to use somatic mutation fingerprints as predictors, as the features, and then predict cancer types or specific cancer risk factor outcomes as the responses. There are some key challenges, though. The main challenge is that somatic mutation data is extremely sparse and high dimensional. So because the DNA is so long, so big, there are billions of places where alterations can happen. And so if we are trying to build a predictive model, we are talking about millions, possibly billions of positions that could cause cancer. So we are talking about building a classification model where we have more than millions of positions where alterations can possibly happen, and we have different cancer types as the response. For example, in this is one of the data set that I'm going to be focusing later on in my talk. So this publicly available pan-cancer analysis of whole genomes data set, where we focused on 10 common cancer types, most commonly occurring in the US. They discovered in about 1,700 tumors more than 30 million mutations. So from 1,700 tumors, they cataloged more than 30 million mutations. More than 99% of these mutations are called singletons. So they only appear in one patient. There are a handful of recurring or hotspot mutations, very specific mutations that are highly predictive of different cancer types, but that's only a handful. It's a tiny subset of all the mutations we have collected data on so far. And on average, the most challenging part is that on average, more than 50% of mutations observed in any new tumor are previously unseen, previously unobserved. So we are talking about doing a prediction, and then in a new tumor that appears, we do the genome sequencing, look at its genomic fingerprint, and we encounter mutations on which we have no prior data. We have not seen that mutation before. How to build a classification model with such data? So I will not get into the technicalities, but just purely for the sake of building the foundation, I will treat CN as the cancer types, little n for a generic entry, and capital N for the total number of tumors. I will have a predictor matrix X and J. So those are going to be the binary 0-1 indicators of Jth mutation in the nth tumor. And the aim is to build a regression or we will try to build a soft classification problem in a regression framework where we will have these CN, these cancer labels, as our response outcomes based on the predictors X and J. There are two related goals. One is the clinical goal, which is predicting the cancer type based on this genomic fingerprint that we see for a new tumor. That's the prediction goal. The other is inferential goal, interpretable inferential goal, which is understanding how these different mutations affect the prediction, understanding how these different mutations cause different cancer. That's a scientific understanding goal. Two related, but a bit, the focus is a bit different. So when doing this work, we recognize there is an analog in NLP, in natural language processing, and that's for text classification. We're interested in classifying a bunch of text documents based on their word contents, with the analogy being the document, each document is like is tumor, like each tumor is like a document. Mutations are like the words, and document classes are the cancer types. Document classes can be like sports, economics, politics, entertainment, etc. Based on the word content, we want to classify a new document into one of those categories. Now, as we humans do, when we are reading text, we encounter new words. The way we try to infer the meaning of the new words is look into the context. What context does this new word appear in? We try to guess the meaning of this new word based on the context. So the analogy here is called this context-based learning, to trying to infer the effect of a new mutation based on the contextual information we might have on those new mutations, new or rare mutations. Directly on those specific rare or new mutations, we do not have enough information, but possibly based on the contextual information, we might be able to infer something, might be able to say something about those mutations. So we call these meta features. So features about the mutation themselves, meta information about the features themselves, we call it meta features. So for our study, we focused on three specific types of meta features. One is whether or not the mutation, whether it's new or previously observed, whether or not that appear in a well-known list of cancer genes. So basically, these specific genes have been previously studied extensively and have been recognized that they are strongly linked with cancer. So we're going to ask the question, okay, this new mutation, does it appear in one of those about 600 cancer genes? The second is type of alteration. So there are 96 possible types of single base substitution categories. We're going to try to understand this mutation, what type of nucleotide change does it connect with? Does it associates with? Finally, the topological position. Where in the chromosome does this mutation occur? So when we see a new mutation, or possibly a previously observed one, but only a few times, a handful of times, we're going to ask these questions. Okay, is it a cancer gene? Is the mutation at a cancer gene? Is it which of the 96 different categories of mutation does it associate with? And then the position on the chromosome. Mathematically speaking, we basically, from those millions of mutations, we are now looking into a space of much smaller dimension, 3,700. So we are asking questions that has, those are categorical answers into the dimension of like 3,700. So, and mathematically speaking, so I'll be talking in terms of a simple statistical model. We can, of course, make it more flexible using more modern machine learning methods, but the simple models help intuition. So that's the motivation for this talk. A simple enough statistical model where we have the multinomial logistic regression's multivariate counterpart. We have these different cancer types, that's the response, and we have these different mutations, those are predictors. We are building a multinomial logistic model. And what we are doing here is the main step, the key step is the middle one in the three-step multi-level model. The middle one, this beta JK, that line is basically the key context-based learning step embedded within the model. What it's trying to do, it's trying to infer the effect of a specific mutation J in cancer type K. It's trying to build that regression coefficient as a meta feature regression itself. So it's looking into the contextual information encoded in this vector uj, and wk is the effects of those meta features. So it's trying to model the individual effects of the observed mutations and unobserved mutations via the meta features. So that's the context-based learning. That is the key step that is helping us infer through the context. The final step here, those are Bayesian multi-level models. Those are there to help with sparsity in the data to ensure regularized estimation. The main benefit of doing this is that once we write this model, do a little bit of algebra, we will see those whole 30 million dimensional vector of 0, 1s, indicators of whether those 30 million observed mutations happened or not. Once we put it inside this modeling framework, we're going to get reduced quantities like this, xn transpose u. U is the vector of these contextual, indicators of contextual information. Once we do this, it's basically going to be the number of mutations, count of mutations happening at different contexts. So we are going to get reduced quantities like the total number of mutations happening at gene 1, total number of mutations happening at gene 2, etc. Similarly, total number of mutations happening at the first chromosome position, second chromosome position, etc. So it reduces from xn, this xn transpose u via this model, this reduces a 30 million dimensional mutation indicator vector into more like a 3,700 dimensional contextual information vector, which is the key statistical and computational efficiency step that we get from this. So once we do this dimensionality reduction via this model, we are able to fit this model using existing software. We do not need to do very fancy computations here. Just straightforward existing tools for point estimation can be efficiently implemented using existing theory, and that approach does provide scalable and accurate prediction. I will just highlight with a particular example on the publicly available data I previously noted, where there are about a little over 36 million mutations, of which 35 millions were observed only once, were singletons. So very sparse data. On this data set, we applied the model and we looked into how much information we get by this dimensionality reduction. And we were able to see that at the whole genome level, so this is a heuristic visual of the dimension reduced xn transpose u, those vectors. We are doing this dimensionality reduction t-SNE to do this plotting, embedding that 3,700 into a 3D approximate embedding plot. And we're seeing here that at the whole genome level, those xn transpose u basically clearly separate these different types of tumors. So the cancer-specific information is there's lots of cancer-specific information at the whole genome level. This is at a whole exome level, not as much, but still some separation. At a targeted cancer gene panel level, the information is nearly not as much as the whole data. So this gives us some insight on how we could extract information from the whole genome panel. But at the end of the day, this is a simplified statistical model, so you could still look into individual effects of these different parts in the model, different genes, different mutations, individual mutations, different types of alterations, etc. It's a statistical model. You could look into the odds ratios and do statistical inference with this, even with all this high dimensionality. So this approach permits scalable estimation and prediction. It can extract and reveal informative cancer-specific information. But the main challenge with this that we were not able to solve for the longest time was uncertainty quantification. So in this framework, we could not do UQ because it's so high dimensional, the predictor variables are so related, and We could not do a full Bayesian inference. That's where we took another inspiration from the field of NLP, topic models. Again, these are all areas in NLP that has been there for quite a little while, not as new as LLMs, but these also translate directly into our framework. In the genomic context, so we have all these different types of mutations or contextual information, which are directly not particularly related, like individually they are noisy, but under the hood, there's some meaning attached to them. Just like in topic models and with words appear somewhat randomly, but they do have some meanings in text documents. There's some latent semantics, similar meaning words. They're called topics. So the same idea that we employ in this framework, the formal name for topic models is called mixed membership models. So we utilize them within our statistical learning framework to do further dimensionality reduction for those mutation types. Without getting into the details, the key step is we embed this specific type of topic model-based dimensionality reduction technique within our model so that we could do full Bayesian inference for uncertainty quantification. So we employ this topic model layer, one particular version of that, which is called non-negative matrix factorization, NMF, which is a highly researched topic in computational genomics. We embed that into this framework and connect it within this multi-level modeling context. Here's a very high-level schematic for what the model is doing. We have on the top left, we have the individual mutations like text documents that collects those mutation information. And via these meta-feature learning and then topic modeling, we are connecting it with the different types of cancer. So here's a bit of technical details on how we do this final inference from the model using Bayesian computation techniques. Not going to get into the technical details due to time constraint, but as it would turn out, our model is related with the main topic of this work is doing mutational signature analysis. And as it would turn out, embedding the topic model would result in structures that are essentially identical to mutational signatures that has been investigated in depth in computational genomics. So here I show two topics, genomic topics that we discovered from this, and there are high similarities. There are large similarities of these with the traditional mutational signature that is discovered in computational genomics. The main difference being our approach is more supervised. It's like cancer type information is embedded within this framework. So those are directly informing us which mutation topics to look for, which mutational signatures to look for a specific type of cancer. And it also helps us do uncertainty quantification in this framework. There are several challenges in this that we are yet to resolve. Is prediction is not up to the mark because it's a simplified statistics model. We experimented with integrating this into a more tree-based classifier for improved prediction performance. And long story short, we were able to see noticeable improvement in prediction by integrating a random forest within the framework of this multi-level prediction model. So if we are able to do more intense computations using more flexible models like deep learning, we should be able to get more informed prediction within this framework. So as a summary, the key take-home messages from this area of work is that leveraging mutation contexts can permit highly informative signal condensation for prediction as well as inference. Topic-based dimensionality reduction taken from the area in NLP can help us give interpretable structures or patterns of mutational topics, analogous to mutational signatures. But there are lots of directions, lots of future work. So first, we will have to formalize this MCMC-based Bayesian modeling for inferring the topics, more scientific investigation into different types of topics, particularly the chromosome window topics, and integrating other risks and clinical outcomes within the framework. So I will stop here. Thank you very much for your attention, and happy to take questions.
Very on time.
Thank you so much, Saptarshi. Any questions from the audience?
Yes.
Question.
Yes.
So much for this great talk.
Thank you so much for the great talk. Two questions. The first one is, you have this topic modeling, right? You validate against existing findings. Looks like it's very much aligned. So what a new discovery have you find from the topic modeling? That's the first one. Yeah.
That's a very good question. So we are indeed, we have been able to identify a few interesting topics, but I did not show it because we have not yet been fully able to validate it. Because, to validate it, we will need to have independent data sets. Like our research is primarily based on publicly available data set. We did find interesting findings. We are trying to do secondary validation for them using their associations with other genomic variables. But direct validation based purely on computation model is tricky. What we did do here is we were showing that our methods produce some interesting novel topics and some existing, like a reproduction of existing topics, which are more like proof of principle, but with attached uncertainty quantification.
Okay, thank you. The second question is, like you have this mutational data, right? That's all the information you use as also as a context. But I don't know if I'm not this domain, so I don't know. Okay, how do you take into account the structural information, which I heard is also very important for gene expression, right? So,
absolutely.
Yeah.
It's a very insightful question. Yes, indeed. So for here, we are using those contexts to partially provide us with the structural information, like specific genes, where in the chromosome it's occurring, what type of nucleotide change, but certainly other types of structural information, like information about methylation and methylation and like possibly structural variants. Those can further inform. The three-dimensional structure, it's indeed, like I'm getting into that. So the 3D, the problem is that our methods are limited to the data we have. We are using publicly available data, and there's not enough info about the 3D structure just yet. We're looking into new investigations, large-scale investigations that are making the data available. When we get those pieces of information about 3D structure, it would be somewhat, it would be somewhat, I would say, straightforward once we have those encodings integrating into the model. But the main challenge is getting the data. Once we get it, we are going to be absolutely, psyched to put it there. Thank you.